So we might as well start with the introduction so that we can then start on time. It's 59, right. So welcome to this session. It's the Symposium 53, Tropical Ecology, and the part Ecosystem Processes and Function. As you can see, I'm Malte Jochum, the replacement for Andrew Barnes, who was supposed to chair the session and would have really liked to do it, but unfortunately he got sick, so I'm the replacement and will do my very best to guide us through it. So the first speaker is David Burslem, and he's going to talk about aluminium accumulation in tropical forest plants. And now I guess we need to bring this up. Right. Come on. Okay, that's it. And F5, and you're ready to go. I guess that should work.
Yep.
Awesome. Good luck.
Great. Thank you very much, Malte, and thank you for making your way after the lunch. So my task, I want to introduce you to what might seem as like a slightly esoteric group of plants, but hopefully I'll be able to convince you within the next 15 minutes that they are important, they're abundant, especially in some of the floras that we work with in the tropics. Possibly the aluminium accumulation, we interact with it most frequently when we consume tea, because tea has a very high concentration of aluminium in its leaves, as many of you may know, usually cultivated in this kind of environment. Perhaps familiar with this sort of landscape in some tropical environments. As a digression, I would just point to the fact that we can grow tea in the UK. So there's an increasing UK. There are 13 growers now, including some in Scotland. Tends to be quite a high-priced product, but for tea lovers, it might make a great Christmas present. But aluminium accumulation, despite having, appearing to having been described a very long time ago in plants like tea, it turns out it's actually very important, very significant in terms of the numbers of species that do that. So 45 angiosperm families and indeed many other kinds of plants have been shown to include species that accumulate aluminium, or at least have high concentrations of aluminium in their tissues. That represents about 5% of all angiosperms, so possibly a higher frequency than many of us might have considered. And if we look at those list of families where it's especially common, so the Simplicaceae, Melastomataceae, Rubiaceae, and so on, those of you who inventory tropical forest floras will instantly appreciate that those include the families that have numerous species in the understories of wet tropical forests. And indeed, if we go into some other kinds of ecosystems in the tropics, so thinking about savanna systems in Latin America, for instance, there are very, very high frequencies. Almost 100% of the basal area of trees in those systems would be in families like the Vichiziaceae, for instance, where almost all the species accumulate aluminium. So that's an indication of its importance, and especially in tropical tree floras and the understory, the species-rich understory of tropical wet forests. We're within a few months of actually finishing an inventory where we've systematically sampled every tree and where we might be able to actually quantify that. But we think it's going to come out at approximately 50% of the basal area of understory trees in some lowland tropical forests in Sabah, for instance, will be species that accumulate aluminium, will have the capacity to do so. Okay, so these are the kind of the questions that have interested us over the last few years. So firstly, taking a look at the distribution of aluminium concentrations in plant tissues, is it simply a statistical artifact? And I'll come back to that in a moment. And then sort of secondly, why on earth are plants doing this when we know that aluminium is a metabolic poison, both for plants and also for animals, of course? So there's a functional question. And then there's the more mechanistic question, the physiological question: how are the phytotoxic effects of aluminium mitigated in plants, so how do they cope with it physiologically, and then scaling up what would be the ecosystem consequences of aluminium accumulation. So then just to address that first question, the sort of motivation for this is, if we think of any ecological trait, then there will of course be a distribution of values among species, and foliar aluminium concentration would be no other. So in order to consider aluminium hyperaccumulation or accumulation as a distinct entity, as something that's just a discrete trait, we need to think about whether the accumulators represent simply the high tail of a distribution of values, or in fact are we, so the situation we see there on the left-hand side, or perhaps is there any evidence that the distribution of aluminium concentration in plant tissues is bimodal, in which we can identify a group of species that are definite accumulators and they're distinct from another group which are non-accumulators. So we believe that aluminium accumulation is a distinct entity if we find evidence of the situation on the right-hand side. And to answer that question, we've reviewed as much information, as much data as we could find in the published literature on aluminium concentrations in plant leaves, which is the tissue that's most commonly measured, of course. And we've compared those distributions to that of other elements, in particular the nutrient elements. So here are some frequency distributions of elemental concentrations. So from nitrogen on the top left, aluminium on the top left, and then 5 other nutrients that are commonly measured in plants. And these are simply just harvested from the literature. And it's clear that if you look at values for hundreds of species, for nitrogen, phosphorus, and the cations, what we observe and what we can statistically verify is that these are simply normal distributions. There's no evidence of bimodality in these frequency distributions, whereas for aluminium it's very different. We do find there what appears to be a bimodal distribution, and the analysis validates that. There's significant evidence of bimodality in that frequency distribution. So we conclude from that there is a thing that we can regard as aluminium accumulation, and that we can identify particular species of plants that have that trait. So what are the potential reasons why plants might accumulate a toxin in their tissues? Well, perhaps they're doing it simply because they have to. They're growing in aluminium-rich soils. They're simply passively taking it up. We might be skeptical about that because we find trees of very closely related species in the same genus growing side by side in the same soil, which do and don't accumulate aluminium. So there seems to be a capacity for very closely related species not to accumulate aluminium, even though their congeners do. But that's a possibility, of course. There are some anecdotal sort of records in the literature that some plants, and petit is a good example, which actually respond positively in terms of their growth to aluminium. So we've been interested in that and testing in it, looking at what the fundamental physiology of that might be. And then there's the possibility, I suppose, that that aluminium concentrations in plant leaves are a defense against something, perhaps fungi, perhaps insect herbivores, because of its toxic properties. Okay, so we can address whether foliar aluminium concentrations are non-randomly distributed across different soil types when we look at a range of species, and we've used Brunei as a good testing ground for this, because we see forests growing on very different soils in close proximity in Brunei. And 2 particular kinds of forest, heath forest and lowland mixed apricot forest there, have very different aluminium concentrations in the soil. So that the table in the bottom there shows values of soil aluminium, both measured as total concentrations and also the exchangeable fraction, which are significantly higher in mixed apricot forests than they are in heath forest soils. And that's well known, and it's something we've been back to look at as well. And then we look at the habitat specialization of trees that occur in both those kinds of forests, both the specialists of mixed apricot forests, the specialists of heath forests, and the generalists. And there clearly does seem to be a signal of soil aluminium concentration in the foliar aluminium concentration. So those are box plots of foliar aluminium concentration on a log scale, and the values of mixed dipterocarp forest trees are significantly higher than those of the heath forest species. So there tends to be an association there. So perhaps there's some evidence that you're more likely to be an aluminium accumulator if you grow in an aluminium-rich soil. We wanted to test that at high resolution, and for that we made use of the fact that this particular plant, Melastoma malabathracum, is extremely common and widespread across Southeast Asia and grows in multiple different kinds of habitats with different soils. Some of you you must recognize it if you've ever spent any time in Southeast Asia. It's a very common plant in the Melastomataceae, of course. So we sampled Melastoma from a range of different sites, so multiple populations across Peninsular Malaysia, distributed there from all sorts of different habitats, including montane, freshwater, coastal vegetation, and so on. And measured aluminium concentrations in leaves, and then we also collected seeds, grew up seedlings in solution culture, and supplied them with aluminium and looked to see whether they also accumulated aluminium. So we simply ranked them there according to the aluminium concentrations in the seedlings that we grew in solution culture with aluminium supplied. And then the grey bars are the equivalent concentrations of the adult plants growing in the field. And you can see, for one thing, firstly, of course, there's huge variation in aluminium concentrations in both groups, both the adults and the seedlings. All of those populations expressed concentrations that are above the arbitrary threshold of 1 milligram per gram to define them all as accumulators, but they accumulate to very different degrees. So again, we wondered whether this was related to the fact that they were growing in soils that supplied them with very different amounts of aluminium. And the answer to that is a definitive no. There's no relationship whatsoever between aluminium concentrations in leaves, either of the adult trees or of seedlings when you grow them up in a common environment, and the aluminium concentrations in the soils from which those individuals came. So that's interesting. However, there does appear to be an association between aluminium concentrations and the concentrations of other elements in soils, particularly the cation, the macronutrient cations, calcium especially, and a positive relationship between aluminium and calcium concentrations in the leaves. I want to introduce you finally just to this phenomenon of beneficial effects of aluminium on growth, because it's such a paradox. Why plants would grow faster when you supply them with something that is ultimately a toxic, a metabolic toxin. And we've verified that both in terms of dry mass of seedlings and their relative growth rate. Again, these are experiments in solution culture, and you can see the picture there sort of provides a visual representation of the size of plants at the end of an experiment where you grow them with very different concentrations of aluminium in the nutrient solution. We've been interested in what the physiological drivers of that might be. And interestingly, and this is the key point here, at least for some populations that we collected from Peninsular Malaysia, there's also a positive effect on photosynthesis. So it seems to be that supplying these plants with aluminium results in an upregulation of photosynthetic rate and of respiration rates. And we wondered whether that would be associated with other aspects of carbon partitioning in the plants. And these are data hot out of the lab from last week. My PhD student, Khairul Mahmood, has shown that that's the case. So when you supply these plants with aluminium, they upregulate photosynthesis, and a lot of the carbon that's generated as a consequence of that is transported to the roots, and there it results in an increase in sugar concentrations in the root system, and that's associated with higher non-structural carbohydrate concentrations in the entire plant as well. So there's something very strange going on here, and we think it's probably related to the fact that these plants need lots of carbon in order to transport aluminium from the roots to the shoots. And so they transport these non-structural carbohydrates, the sugars, to root systems in order to allow that to happen. Okay, so I'll end with some conclusions that I'll allow you to read, and I know the chair would like us to move on. Thank you very much.
Yeah, thanks for the very nice talk. I think we would have time for a very quick question while I already changed the slides. So if you have one, go ahead. If not, then I'm asking, like, how do they store the aluminium? Do they like?
It tends to be stored in cell walls and in vacuoles, so it has to be stored away from the metabolic places where plants are sort of where they would otherwise cause negative effects.
And is it just in leaf tissue or is it everywhere?
It's everywhere, yes, indeed, and even in bark you can identify high concentrations. Yeah.
Thank you very much.
Oh,
Sorry.
we'll meet after.
Okay. Right. Our next speaker is Wenjing Liation and temporal variation of tropical mangrove leaf litter fall.
Okay, hello, thank you for coming. So now I'm going to bring you to the coast and to these fabulous mangroves of tropical areas. And I'm going to talk specifically about litter fall. But why do we study litter fall? Actually, it's been claimed that mangroves are very important carbon sinks, especially amongst coastal areas. And this observation, and actually the coining of the term blue carbon to represent all this big stock of carbon in coastal areas, these observations come from the evidence that actually mangroves have a much a big carbon stock in their sediments, especially when compared to other coastal areas. And all this carbon, all this accumulation of carbon in the sediments is a result, a combination of litter fall rates, exportation to the adjacent ecosystems, and local decomposition rates. And what is it that we know so far about litter fall in mangroves? Well, we know that they represent around 57% of the above ground NPP, so more than half of the 1 year production is actually shed and incorporated into the sediment. Especially in tropical and equatorial areas, litter fall can be as high as 26 tons per hectare per year, which is a lot. And we know that, well, this is not very nice, but from 30 to 90% of this litter fall might be exported to other systems. And we also know that it varies a lot seasonally, as you would expect, of course, because the plant physiology is also being regulated by differences between the seasons. What we don't know, it's about species-specific patterns of litter fall, because most studies, they focus on the actual biomass and the actual production, and they don't focus much on differences between the main species or the dominant species. And we also don't know what explains the sources of spatial variation that can be actually very high, because most studies only correlate litter fall with fluctuations in climate. So this is what I try to do here. I try to focus on these two aspects, species-specific patterns and spatial variation, and to see what else can we learn, what else can we derive from litter fall patterns that we don't know yet. To do that, I focus on these mangroves in the north of Brazil. This is a very interesting study site. It's been studied by ZMT, the institute where I work. They did extensive series of studies there in the 90s and early 2000s, so there's a lot of data on litter fall, on vegetation structure, on climate, and I put all of this together, and I'm going to show you this here. So this is my study site, the Ajuruteua in Brazil. And these mangroves are composed by three main species: Rhizophora, the red mangrove, Avicennia germinans, the black mangrove, and Laguncularia racemosa, the white mangrove. But for this presentation, I'm going to focus on Rhizophora and Avicennia. So the meta-analysis, I included in this study only studies that reported monthly species-specific leaf litter fall, and I only included studies that presented dry weight per unit area, that used litter traps of 1 square meter, although the sampling effort was different between the studies, minimum of 12 consecutive months, and for studies that were longer than 12 months, I treated each sequence of 12 months as a replicate. So in the end, I found 6 studies that studied 11 sites, and with 20 replicates for Rhizophora and then 24 for Avicennia. So you have an idea, this is the mangroves, this is the peninsula, and for all the green sites, I had vegetation structure. So diameter, height, basal area, density, and dominance for Avicennia and for Rhizophora. And for 11 of these sites, I had data on litter fall for Avicennia and for Rhizophora. Okay, so this is the overall results. You can see that in most of the sites, production by Rhizophora is much bigger than by Avicennia. And it ranged, and here I'm focusing only on leaf litter fall, so we ranged from 0.4 to as much as 10 megagrams per hectare per year. So I correlated the vegetation structure to the litter fall patterns, and using a PLS regression, and I got actually a very good fit. So for Rhizophora, I have 60% of variance explained, and for Avicennia, 81. And I was very happy because having this in the model, I could do all the other calculations. So what can we do after we have something like this? So this is a PCA of all the sites where I had vegetation structure, okay? And then let me show you something that is very nice. So here, so for black is Avicennia, the black mangrove, and in red we have the red mangrove, Rhizophora, okay? And if you see these sites here, they represent sites of high salinity, where Avicennia is really, really abundant, but the trees are very, very short. They can be dwarfs of less than 1 meter, but densities is like 90,000 individuals per hectare. So this is one type of physiognomy you find there. And then here, in sites that are not so stressful, we can see that Avicennia can grow much more. And then, so we have here lower density, lower diameter, but a higher trees. And when you move from here to there, you see the older sites where Rhizophora starts to dominate the sites, and then Avicennia is outcompeted by Rhizophora. So the few Avicennias that are left, they are tall and they are big, but there's only few of them. And the sites are dominated by Rhizophora. So this is the conditions of the vegetation. So what I did was I simulated, and these are the sites where I actually have data. So what I did, I used multivariate correlation to simulate sites with intermediate characteristics between this, which will be very, very early succession in the mangrove, to that, which will be the more mature, more older mangroves. And then for each of the simulated sites, I calculated with my previous model, I calculated how much leaf litter they will produce. And what we have is this. So from pink to green, we have increasing production of litter fowl, and this is the sum of the two species. So when Rhizophora starts to appear, and then you can see here. So from the transition from Avicennia-dominated sites to Rhizophora-dominated sites, we will have a very big increase in litter fowl production. And this is interesting, especially if we are interested in carbon stocks and decomposition rates, because Avicennia and Rhizophora are very, very different from each other. So Avicennia has tougher and thicker leaves. It decomposes faster and is richer in nutrients, as you would expect from a more pioneer species, whereas Rhizophora has much more secondary compounds. It decomposes not so fast. It has thinner leaves, and it's very, very intensively used by crabs. So the two types of litter fowl represent very different components in the same site. I also looked at temporal distribution of the, here I also have data for the other third species, and then you can see the species don't shed their leaves at the same time. So this also has consequences to carbon accumulation. So you can see that Avicennia, during 3 months here, between the wettest and the driest month, 50% of all the annual litter fowl is going to fall here in these 3 months. Whereas for Rhizophora, it's more or less constant along the year. And for Laguncularia, it's also very seasonal, but completely the inverse pattern. So the adaptation, the response, the physiological response is not the same to the seasonality. And here another example of what we can do once we have data like this. So I combine the sites. So each line is one site. I combine the sites with the temporal distribution of the litter and the amount of distribution of the litter, and then how much they have in carbon and nitrogen, and then we can see during the year and in space where you have higher input of organic matter. And to conclude, so yeah, as you see, we in the study site, the leaf litter fowl patterns are species-specific, and we can predict them based on vegetation structure. Rhizophora-dominated sites produce more leaf litter than Avicennia, and this has consequences because the quality of the organic matter changes along succession and also changes during the year. That's it. Thank you. It's not into the model, it's two independent, okay. So this is the okay. This is the simple PCA with all the sites and the height, diameter, density, yeah? So once I have this PCA, I simulated new sites according to the multivariate correlation between these parameters. So I simulate them. I take out of the simulation all the sites that by chance have values that were not higher or higher than what we observed. So I had to trim a little bit. And then for each new site, you plot on the existing PCA. And then for each site, I put the data for each simulated site back in the model that I had before, and then I can calculate how much litter fall for each species, and then summing this. Is that okay? Yeah.
I also have a very quick question. You said that one of these, for one of these species, the leaf litter is used by crabs.
Ah, yeah,
What do they use it
Hisophora. They love it. They eat
They
it.
eat it.
They eat it, and then it's very interesting actually because the crabs love Hisophora, and they tend to concentrate under the roots because they are more protected by predators. So they make their burrows, and then they collect the leaves, and then they put it in the burrows, and then they compose that, and they eat them. So for this site, it's estimated that the crabs consume 60% of the annual litter fall, but only for Hisophora. They don't like Avicennia. So they probably help capturing the leaves inside the burrows, so these leaves will be decomposed locally, whereas for Avicennia, probably the water will wash them away. And
Thank you very much.
thank you.
Right. So the next one is, it should be 30, 45. Frozen. It's not here. Maybe it's this one. Oh no, that's the session sign. Is Geertje van der Heijden here? No. So the next speaker is Geertje van der Heijden.
She's at the program. I haven't seen her today. She's not been around.
Okay. I just discovered that the slides aren't here either. So
We can try and give her a call.
yeah, sure. I mean, otherwise we have a no show. I mean, I can juggle something if you want, but. Sorry about that. Right, so we'll just wait. Doesn't answer the phone. Right. So we can't shift talks. I guess that means that you either have a nice break or talk to the last speakers, or I don't know. Right. So have a break. Thomas, and she's talking on ant diversity as a driver of beetle functional diversity.
Hi. So I'm going to be presenting my master's research that I did with the soil biodiversity group at the Natural History Museum. So we looked at so we focused on myrmecophily, which is a unique biological association that forms between ants and non-ant species, and that can be seen across many different insect groups, but there is an evolutionary bias towards Coleoptera, where there's an estimated 10,000 species of myrmecophiles. And within this order, the family Staphylinidae, which are commonly known as the rove beetles, have the most number of myrmecophilous lineages, and within this, then that includes the cellophines, which is the focus of my study. So cellophines are a species-rich predatory leaf litter beetles found in both temperate and tropical rainforests, but more dominant and diverse within the tropics. So in Borneo in particular, there's 123 species of cellophine beetles, but although that's quite a small number, so there's still many more to be described and recorded in Borneo. So cellophine ant associations can vary from facultative or opportunistic nest intruders. So this is a casual association that forms where the ants occasionally tend to the myrmecophile guest, or it can be obligate myrmecophily. So this is where the beetles are entirely dependent on the ant host, and they actually live inside the ant's nests as well. So cellophines are also habitat specific, so they inhabit moist leaf litter and woody debris microhabitats. And so in addition to that, they're still very ecologically successful within leaf litter invertebrate communities. And so it's of great value to us to try and understand both their ecology and behavioural interactions, particularly in response to ecosystem disturbances in vulnerable habitats. So one way to do this is to try and understand their morphology. So a typical staphylinid body plan is characterised by 11 antennal segments and truncated elytra, and they have quite an elongated abdomen with 7 visible abdominal segments, which is quite flexible for them. But the cellophines deviate from this typical staphylinid body plan, so they're much more compact and sclerotized integument, and there's only 5 visible abdominal segments, so it really restricts their flexibility, or their abdominal flexibility. So this is just a selection of a few species of cellophines, just to show their diversity, and in particular you can see their antennae as well are greatly diverse in structure and shape, and you can see sort of in the top left one as well, they have this unique club formations which can vary a lot as well, compared to the one on the right here, which has this large block-like antennal club. But the modifications are most extreme in the obligate myrmecophiles, so in the tribe Clavigerini, which you can see here. So they have greatly reduced number of antennal segments, and also the abdomen as well. So there's great fusion in the abdominal segments, and they actually form this deep basal depression, which you can see there. Okay, so the aims of this study was to measure the functional traits of cellophines that were specifically associated with myrmecophily. So one, to allow us to understand how cellophines interact with ants within the leaf litter, but also inside the ant's nest as well. And two, how these functional traits can then be applied as a measure of functional diversity in ecosystem disturbances. So we predicted that the functional diversity of cellophines will increase in the presence of both ant abundance and ant genus richness. So our study sites, so we use primary rainforests, so in Malaya Basin and Danang Valley conservation areas in Sabah, Borneo, and the logged rainforest site, which is part of the SAFE project sites there. So we collected the leaf litter invertebrate samples using the Winkler bag. So it's basically sieving 1 metre square quadrats of leaf litter along a 100-metre transect. And then we identified the celephines, first to morphospecies, and then to tribe, and then genus. And the ants were also collected from the same samples as the beetles. So we selected 11 functional traits to be associated with myrmecophily, and this was mainly based around sort of 4 main areas. So the first being the presence of trichomes. So trichomes are thick brush-like setae, which secrete glandular exudates, which are attractive to the ants. And this initiates trophallaxis, which is a unique liquid mouth-to-mouth feeding behaviour initiated by the ants to the beetles. And then also we looked at the fusion of antennal and abdominal segments. So this increases the strength, it strengthens the body for protection against handling by ant workers, and also increases the surface area for secretions to spread. And lastly, we also looked at, thirdly, we looked at body size as well. So an increase in body size has been linked to inquilinous lifestyle, potentially due to the nutrient-rich environments that ants' nests provide. And lastly, we looked at foveal patterns. So fovea are small pores sort of in the exoskeleton of the beetles, both on the dorsal and ventral side. And although the actual functional role of these fovea are not really known, so there's been 2 proposed suggestions: one, that they serve as a sensory function, and 2, that they provide structural rigidity. But they haven't really been tested out as much. And also it's quite interesting is the fact that even though they don't really have a direct role within myrmecophily, there is an evolutionary trend towards loss in the number of fovea present in myrmecophiles, so that's something we also looked at. So we measured the functional traits, and we took lots of images of the beetles as well, to try and, to measure the different traits, and also to try and identify some of the traits as well. So we did some SEM images to try and identify particularly the fovea, which you can see sort of in the top middle photo at the top there, which otherwise we wouldn't be able to see under a normal microscope. And also to look at the structure and shapes of the trichomes, but also just to try and identify any other additional traits or any interesting features of the beetles that we otherwise wouldn't be able to see. So then we applied our functional traits skipping ahead there. So we had a total of 613 celephine beetles across all 3 sites that we sampled, and which represented 12 different tribes, and there were 42 morphospecies in total. And of those 42 morphospecies, 12 of them were 12 belonging to 6 different tribes, were recorded as probable myrmecophiles, which you can see here. So the top 2 at the left there belong to the Clavigerini, which is the obligate myrmecophiles, and then the other 5 tribes represented by the other morphospecies here, and they all form a facultative association with the ants, so they're quite varied there. So then we applied our functional trait data to measure functional diversity. So here, functional diversity is an estimate of myrmecophily. So we can see that myrmecophile functional diversity is significantly correlated to ant abundance and ant genus richness. So we can say that the presence of myrmecophile functional traits increases in the presence of increase in ant genus richness and abundance as well, which agrees with our predictions. So in conclusion, we can see that there is an important link between adult morphological characters and the functional roles that they play in myrmecophily. And it's plausible to say that ant diversity is, or maybe a direct driver of celephine beetle functional diversity. And so we can gain a bit more of an understanding of their ecological interactions within the leaf litter, which is good. So it offers a lot more potential, I guess, to find out more about their roles and functional roles within the ecosystem, particularly in ecosystem disturbances as well. So yeah, thank you.
Thank you very much. That was quite
It's
fast.
quite quick,
So,
yeah, sorry.
Yeah, hope we have some questions. Right.
Two quick questions. Do you know if there are particular ants that beetles are associated with, and what does it mean?
Yeah, so they're predatory leaf. So sorry to answer your second question. They're predatory leaf litter beetles. So they typically, I guess, are the smaller invertebrates like mites within the leaf litter. And sorry to go back into your first question. So there have been some cellophane ant associations that have been observed directly out in the field, and they've been with Lasius ants. And that's been, there's a few studies, or there's a group that's worked on them. They've been collected from northern Thailand and some in China as well. I think there's probably a total of like 5 studies that have reported that, so it's still quite scarce within the literature, but it's mainly from Lasius ants. So, yeah.
What's the nature of the relationship between the ants and the beetles? I know you mentioned something about the feeding.
Yeah. In terms of what's the...
Well, yeah, I mean,
Where you
so
said
the
the
ants are feeding from the beetles?
Yeah, so in the obligate myrmecophiles, they so it's referred to as trophallaxis, so that the ants, it's mouth-to-mouth feeding from the ants to the beetle, and it's they just feed them like this. It's a liquid
And
compound.
are the beetles then getting protection from the ants, or are they just
Yeah,
scavenging with ants disturbing?
Yeah, so I guess their nature is quite, it's referred to as social parasitism. So I know myrmecophily can vary between different insect orders, whether it's mutualistic or parasitic. Within Coleoptera, it's referred to as social parasitism. So it's, there appears to be no direct benefit to the ants, and it seems to be more that the beetle just gains from them. So protection within the ant's nest from extreme weather conditions maybe, and it provides them obviously with food, so they feed on the ant larvae as well. But there appears to be no real direct benefit to the ants.
so
That's
the
kind
ants
of really
let them
been-
feed on their larvae.
Yeah.
They accept that.
So yeah, they seem to be able to withstand that attack from the ants.
Any other questions? Yeah.
So is there any like capacity for co-extinctions or something like that? Or if these the ants going with people as well?
Yeah, so we've, within the research group, so they've done some ant suppression plots as well in the primary rainforest. And we've shown that actually once all the ants have been suppressed, there's been a loss in particular species of cellophines, particularly the obligate cellophines. So yeah, there will be a loss in cellophines.
And is this a study that you might continue to look at how different sort of logging regime functional diversity of the oil palm? There's a lot of work on functional diversity that's gone on down the valley, and
Yeah.
it's really hard to kind of
Yeah, definitely. Yeah, so hopefully I would like to continue this research because I know you looked at primary forest and logged forest, but as you said, yeah, definitely within oil palm plantations as well, and look across the whole ecosystem and environmental land use gradient as well would be really interesting. So yeah, hopefully.
Any more questions?
How did you calculate the functional diversity?
So we use the myrmecophile functional traits, so the 11 that we selected, and then we applied that using Rao's quadratic diversity index. Yeah.
Is that an index of functional dispersion, or does it not have anything?
Not the dispersion, no. Yeah, it just looks at so the pairwise differences, the morphometric measurements, so the functional traits and abundance.
Thanks.
Thank you.
So our next speaker is Laura Allen, and she's going to talk about dung beetle communities across the human disturbance gradient.
Alright, so I'm going to talk to you a little bit about the response of dung beetles to human disturbance in an area of regenerating forest in the neotropics. First, I'd just like to thank everyone who's been funding me and my supervisors and everyone else that's been helping me along the way, just in case I run out of time at the end. And then I guess the next step is to talk to you about why dung beetles. First of all, we know that fruit-eating mammals are really important seed dispersers in tropical forest. However, without any help, the dung they deposit is quite prone to being washed away by rain, and also the seeds in that dung are vulnerable to seed predators, and also face intense competition when they're all trying to establish themselves in that tiny patch. With a little help from dung beetles, they come along and the rollers take the dung and they roll it far away from the original deposit site, and you also have tunnelers who'll be tunneling into the dung. And combined, this helps spread out the seeds so they face less competition. It helps sequester the nutrients from that dung into the soil so it's more available for plant growth, and also reduces the risk of seed predation. And all this tunneling activity also helps to aerate the soil, improving soil quality. So there are two main questions I want to address during this talk. First of all, do dung beetles change across a disturbance gradient? And more importantly, in what way do they change? And does this have any impact on these ecosystem functions that the beetles provide? My research takes place out in Peru, in the southeast. I'm working in the Mangrove Biosphere Reserve. Oop, didn't need to do that. So I work just in this orange zone here, which is the cultural zone. This acts as a buffer for the main national park. And in this area, there's a matrix of different land uses. So it ranges from good forest that's used for ecotourism to areas of agriculture and small villages. In this area, so I'm working at a research station called the Mangrove Learning Centre, which is the red spot over there. And I've identified 18 sites around that area, covering a gradient of 6 of those major land uses. Within this gradient, I've established a rank of disturbance, going from the most disturbed, which are banana monocultures. Then we have agroforestry plantations that include some native tree species, disturbed secondary, which are patches of shrubby secondary forest that have grown up around agricultural areas. Then we have cleared regenerating forest, which was cleared for agriculture, but has been regenerated under protection for about 30 years. Then we have mixed history forest, which was cleared in small patches at different times, but again has been regenerated for about 30 years. And finally, we have minimally disturbed forest, which doesn't show much obvious sign of disturbance, but has probably had a couple of trees taken out in the past. So, I collected my dung beetles using pitfall traps baited with dung, and I also carried out some vegetation collected some vegetation structure data across the disturbance gradient, just to make sure that there actually was a disturbance gradient that was noticeable, which there was. I then looked at alpha diversity of the dung beetles and used linear models to test the effect of disturbance on alpha diversity, whilst controlling for other environmental variables that may have been relevant, and just double checked that there was no spatial autocorrelation, since I'm working in quite a small area. Next, I moved on to looking at beta diversity, looking at how community composition changes across the gradient, how the beta diversity is partitioned, and I also looked at different responses amongst functional groups of dung beetles. So here, as I mentioned, the rollers take the dung and roll it away, and then all the beetles that are just digging around in the dung and burying it, I've classed as tunnelers, even though they could be differentiated a little more if you wanted to. Finally, I did some simple experiments looking at ecosystem functions, which were very much inspired by work by Braga and colleagues, and it involved setting up these small arenas where you clear the ground so you can find all the things that are going on. They limit horizontal movement by the beetles, and in the centre of this little arena we put a pile of pre-weighed dung, and in the middle of that we put beads of different sizes which represented seeds. This allowed us to look at dung removal, which would tell us a bit about the nutrient cycling that the beetles were carrying out, as well as the seed dispersal that they were doing, and a bit about soil aeration by looking at how much soil they were digging up. So, what did we find? Sorry, I'm quite hoarse from all the chatting over the last few days. Well, here we have a graph of alpha diversity. Again, like many people during these last few days, I'm returning to Hill's numbers. Just a few little notes about this graph. I'm going to break it down in more detail over the next few slides. But over at Q equals 0 on the left-hand side, at this point, rare species count just as much as common species towards a diversity value. Whilst over at the right-hand side, only the most dominant species in the community are contributing to that alpha diversity value. Many of you are going to be familiar with these values along the x-axis by their more common names. So at Q equals 0, we have species richness. At 1, we have Shannon diversity. And at 2, we have Simpson's diversity. So, let's have a closer look. So, first of all, at species richness, what we found was that disturbance did have a significant effect on species richness, with much lower species richness found in the more disturbed areas along this gradient. We also calculated estimated richness using Chow-1, just to make sure we weren't missing anything there. So now looking at Shannon diversity, again, we found the same trend here. We found that Shannon diversity was higher in the less disturbed habitats. However, when we move to the right of this graph, we find that there's actually no significant effect of disturbance when you look at alpha diversity from the perspective of Q2, Q3, or Q infinity. So really this emphasizes that it really does depend on how you're measuring alpha diversity as to what effect you're going to see in this dung beetle community. So, moving on, we had a look at beta diversity next. And here, this is just a PCA of how community composition changed across the gradient, looking at which species were present and their relative abundances. And you can see there's a clear shift across the gradient, with more disturbed sites in red here, having very different communities from the less disturbed. When we look at this a little closer, we can see that the majority of this change is due to a replacement of species across the gradient. 63% was due to replacement rather than a change in species richness. We can also look at which species are changing most across the gradient. So which of the species present are contributing most to beta diversity. So of the 6 species here, these were the ones that were found to be most important. Over in the agricultural areas, we have these 2 Canthon species. Monolifer is known to be an agricultural specialist. Virens has been found to quite like open habitats in Cerrado. So fair enough. They like our more open areas. Rubrescens, over on the right-hand side in dark blue, is a forest specialist, and we find it in our forest areas. However, Eurosternus nigrovirens, which is in light blue, has been recorded as preferring intact forest, and yet we found that actually it only liked our agricultural areas. So that was a bit of a surprise. When we looked at the functional groups, we found that there was no change in diversity, with either rollers or both rollers and tunnelers decreased in diversity with increased disturbance, but they showed the same trend. We found no change in abundance of dung beetles overall across the gradient, but we did find opposing trends when we broke them down into rollers and tunnelers. we actually found more abundant rollers in more disturbed areas, whereas we found the opposite trend with tunnelers, where they seem to prefer the less disturbed habitats, which wasn't really what I was expecting to find, as previous studies have found that rollers are more sensitive to disturbance. Finally, looking at the results of ecosystem functions, we found that more seeds were being dispersed in better quality forest, both for small and medium beads. We found that with the largest seed sizes, there was no change across the gradient. However, our other ecosystem functions showed no change across the gradient. There are several possible reasons for this. Partly that it was quite a low power experiment, but also when it came to having lots of different people going out and trying to distinguish dung from soil in quite a wet habitat, that was a little trickier than anticipated. Also, dispersal distance didn't work out quite as well as I'd hoped, because dung beetles reached the edge of the arena quite quickly. We needed the arena to limit our search area, but they reached the edge and then turned around and changed direction, so the end location of the bead wasn't really relevant. So finally, just to conclude, the patterns of diversity you find in dung beetle communities across a gradient of disturbance very much depend on how you measure that diversity. So a holistic approach to measuring diversity seems desirable, or at the very least, a very careful choice of diversity measure. We found that at low values of Q, there was a loss of alpha diversity with increased disturbance, but we didn't find this at higher values of Q. So this suggests that there are higher numbers of rare species in less disturbed forest, but there are similar numbers of dominant species found across the gradient. However, the change in community composition suggests that whilst there are similar numbers of dominant species, these are not the same species, which may impact on other things in the ecosystem. We found some evidence for loss of ecosystem functions in areas of high disturbance, and I think this is an exciting area for further study, but obviously I'm not going to make any huge conclusions from that because it's tentative evidence. But anyway, that's all I have for you today, but if you have any questions, I'd be very pleased, or comments.
So we have a few minutes. Do we have any questions for Laura? I'll just start and join later. So you have this land use gradient there, the intensity gradient. I've been working in a quite similar study actually. So did you validate this intensity? I mean, how do you say that whatever the banana monoculture is more intense than the agroforest? Or is that
I did look at the vegetation structure to see how they changed, but also it was the fact that the banana monoculture only has banana trees and is really, really open. So it is visibly very different and much more affected than agroforestry, because the agroforestry actually had a lot of native tree species there, so at least it did have more structural heterogeneity and
I didn't mean to criticize.
no, we did try and look at that as well, and I mean it's a little bit subjective, but in general, the agroforestry is hoping to try and be more biodiversity friendly, but obviously it might not always be.
Do we have any other questions? Yeah.
It was really cool, it was really interesting. I was wondering, do you have any data on sort of donated body size or like their daily activities? Because looking at that,
Yeah, I've collected data on body size, which I was also going to analyse, like the functional groups, and break that down. I tried to find data on other things like diet preference and activity, but I couldn't get data for all the species, so I haven't done that.
One more very quick question. Can you say anything about the functional redundancy? Have you looked into that by any
I
chance?
haven't looked into that. That would be quite interesting to see how that changes across the gradient.
Do you have the data for that?
I think it's quite difficult, because they vary so much, and I'm very much generalizing with my functional groups, for example.
Thank you very much. Thanks very much. Right. So next one up is Kimberly Simpson, and she's going to talk to us about the influence of fire on grass functional traits.
Thank you very much, and thank you all for sticking around. So yeah, I'm Kim. I'm a PhD student at the University of Sheffield, and I'm going to talk to you today about some work I've done looking into how fire can influence grass traits. Okay, so there's lots of reasons to think that grass may be a fire may be a driver of grass traits. So firstly, for plants, fire is a really frequent disturbance, and one that they've been experiencing ever since they first came onto land 420 million years ago up until modern day. So there's lots of time for plants to adapt to living with this disturbance. Fire is also a really widespread disturbance as well. So fire-prone ecosystems cover a substantial proportion of the terrestrial surface. And fire is also a really severe disturbance as well, and it's one of the few disturbances that will regularly kill mature plants. And for grasses in particular, fire is a really frequent disturbance. So tropical grasslands and savannas experience the most frequent fire of any biome on Earth, and this has resulted in grasses fueling the vast majority of fires on Earth. So there's lots of reasons to think that fire would be shaping grass traits. And the traits that may be selected for under fire fit into a number of categories. So regeneration and recruitment are the two main ways that plants can persist through fire. So regeneration is resprouting from below ground meristems, and recruitment is from the seed bank. Resistance is more associated with woody species. So the picture here is you can see the thick insulating bark of a cork oak. But also for grasses, you get things like, you see in the image above, you get these really tightly packed leaf bases, and they can act as sort of a protection for those below ground meristems. And also flammability, or the propensity of vegetation to burn. And I'm going to talk about that in a bit more depth now. So there are a number of reasons why being highly flammable may be advantageous to resprouting grass species. So firstly, a lot of these species are shade intolerant, and being highly flammable aids in the removal of dead and woody biomass and helps them in that way. Secondly, by burning really intensely but not for very long, less heat can be transferred to below ground and to the vulnerable meristems there. And also, by being really flammable, plants can reduce competition by essentially burning off their neighbours and killing them. So despite these potential benefits of high flammability to grasses, it's a subject that hasn't been widely explored. So there's some basic questions that have remained unanswered. So do grass species actually differ in their flammability? And there's been this historic assumption that they're sort of just a homogeneous fuel to fire. Also, flammability is an emergent property of a plant's chemical and physical traits. But what traits are actually driving grass flammability haven't been determined. And finally, what role evolutionary history may be playing. So can shared evolutionary history explain the patterns of flammability across the grass phylogeny? So myself and some colleagues aim to answer these three overarching questions. And this is some work that was published in Journal of Ecology earlier this year. I'll just talk through it now. So the approach we used was to collect grasses from fire-prone South African grasslands at the time of peak fire occurrence. We built a phylogeny of these species to determine the role that evolutionary history may be playing, and then measured a number of plant traits we thought might be influential to flammability, and also various components of flammability itself, so things like time to ignition, flaming time, and combustion rate. And what we found was that these coexisting species actually differed substantially in the chemical and physical functional traits that we measured. And these in turn drove differences in all of the components of flammability measured. So grasses aren't this homogeneous fuel to fire that had been assumed. We also found that it was biomass moisture content and biomass quantity that were really the key traits in driving grass flammability. And the finding of some strong phylogenetic signals in some of the flammability traits suggests that evolutionary history may be driving at least some of these components of flammability. Okay, so the rest of the work that I'll be talking about today is looking at how fire may be actually shaping traits such as flammability. So fire regimes are hugely variable, and this is true too for grasslands. So they vary in terms of fire frequency and fire intensity. So on the left hand of the screen here, that's typically that's a grassland that typically burns very frequently, and because fuel loads can't build up, it tends to have very sort of low intensity fires. Whereas on the right side, this is a grassland that probably doesn't burn so often, and you get a big build-up of fuel and a really intense fire. So for the rest of the talk, I'll be just considering how frequency and intensity may be shaping grass traits. So I've been lucky to have access to some experimental burn plots down at the University of Fort Hare in South Africa, and using plants removed from the annual burn and no burn plots, tried to answer this question about whether fire frequency is driving trait divergences in 4 savanna grass species. So I removed plants from the 2 treatments, took them back, reduced them to a set number of tillers, and grew them up in a common environment for a year, at which point they looked like this. At this point, I also measured a number of traits. So I measured the number of flowering tillers that these plants had as a proxy for how much they're investing in recruitment as a way of persisting through fire. Also measured their flammability, so I measured their above ground fuel load, and also did some destructive harvests and looked at how they're investing their biomass above and below ground. And after that, I burnt all of the plants and then tracked their regrowth over a month. And what I found was that fire frequency is really driving differences in the traits that I'd measured, such that the annually burnt plants possess quite different traits to the no-burn plants. So the annually burnt plants had 50% more flowering tillers than the no-burn plants, and this suggests that they're investing more of their resources in sort of persisting through recruitment, and that makes sense because the fire's opening up these areas which they can recruit at. Also found that the annually burnt plants were investing a bigger proportion of their total biomass below ground, and this is a trait that's associated with being able to resprout really robustly. And I also found that the annually burnt plants were had a higher fuel load, which we know is a direct driver of how flammable they are. So they're also more flammable. But we didn't find differences in all of the traits measured, so we found no difference in their post-fire regrowth between these 2 treatments. But it did find that post-fire regrowth was positively correlated with fuel load, which again is a proxy for flammability, And so this builds upon some work that some colleagues down in South Africa are working on, that are finding a link between flammability and ecological success in savanna grasses. So flammability may be advantageous because if you can burn off your competition and then actually resprout quickly after fire, you can make the most of that resource-rich environment immediately after fire. Okay, so just the last little bit of work that I'll talk about is looking at how fire, or if fire is as an environmental filter for grass traits, and looking at this at a global scale. So this is the idea that species may be allowed into a population, a fire-prone population, based on what traits they possess. So in this kind of cartoon example, only the plants with pale green leaves are being allowed into this fire-prone community. And I've done this for both fire frequency and fire intensity, but I'm just going to talk about the results for frequency to save some time. So to kind of answer this question, I collected a lot of data from a lot of different sources. So data on species occurrence from GBIF, fire regime data from satellite-derived data sets, grass trait data from a number of sources, and also made the most of an already published large grass phylogeny. And okay, so what you're seeing here, so this is a phylogeny of all the species that had both fire frequency and fire intensity data for. And you can see that grasses do experience a really quite a wide range of these two fire components. You can also see that there is some strong phylogenetic signals, so more closely related species are experiencing more similar fire regimes than those more distantly related. And what I found is that fire frequency was acting as an environmental filter for grass traits at this global scale. So we found that high fire frequency was associated with C4 resprouting species with a low leaf nitrogen content. So, and I'll just talk through these quickly. So C4 species are assumed to have an advantage over C3 species in terms of their greater productivity in fire-prone grasslands, and also their higher water use and nitrogen use efficiency. Resprouting species, we think, have advantage over seeding species, or ones that recruit, because the seeding species may actually be burnt out of populations, but be burnt out of communities because the fire frequency is so high that they're not reaching maturity. And also low nitrogen content. Fire-prone environments tend to be quite nitrogen poor, and so this kind of reflects a high nitrogen use efficiency. So very quickly, just to summarise, hopefully I've convinced you that fire is a really severe recurrent disturbance, particularly to grasses. Fire frequency is driving grass traits relating to persistence and flammability. And fire is acting as a strong driving force, assembling communities based on their traits, and this can be seen at a global scale. And just thinking about the implications of this, we know through climate change and land use change there's going to be changes in fire regime, and hopefully this work can go towards understanding how this may impact on grass communities in terms of their function and composition. Just like to thank these people, and thank you for listening.
Kimberly, we might have time for a quick question if you have one. Yeah.
Thank you. Just to say that it's just a quick comment actually, just so I've got time for that. Common Beetle at York has a massive landscape scale controlled burn experiment in Tanzania, so you might want to get in touch with him, just to see if your traits and data fit with what he's finding.
Yeah, I've met some of his students. Yeah, yeah.
Oh, right.
So yeah, the wonders of ES.
You've
So
already
very
been in touch
deeply.
with us. The behind the curve yet again.
Yeah.
Right, so our next speaker is Richard Dexter, and Richard is going to talk about movement and activity drivers of an ecosystem engineer drive a tortoise. And yeah.
Okay, thank you. Right, so for the most part, over the last couple of days, I've been mentioning tortoise and movement in the same sentence, and I've usually met with looks of skepticism or sympathy. So I'm glad you're all here to help me convince you otherwise. So first up, tortoises are awesome, but secondly, they're really important in any ecosystem they inhabit. They're ecosystem engineers, and like beavers or elephants, they're really important in the ecosystem processes that go on in these habitats. So they do that through seed dispersal, nutrient cycling, and herbivory. And within those areas, the landscapes are quite heterogeneous, and within those areas, resource-rich parts develop, and we call these home ranges. And within these home ranges, we know that the vegetation dynamics occur, and this will change over time, which may force the animals to make decisions to leave to find better resources depending on what they need. So, with that in mind, we go to Aldabra. Aldabra is an atoll that lies 400 kilometers northwest of Madagascar. It consists of 4 islands. Oh, no, that's the wrong one. Where's the pointer? There isn't one. So 4 islands, Picard, Malabar, Grande-Terre, and Pelimni. All of them have tortoises on them except for Pelimni. And the highest density of tortoises can be found on East Grande-Terre, where we have about 18 tortoises per hectare. Conveniently for tortoises and myself, it's very flat, only 8 meters above sea level. And we have quite a variety of vegetation in different mixes in which we can study the system we're looking at. So it's very seasonal. We have very wet summers, very dry winters. And for the purpose of my study, I had to pick what a season would look like. So I've chosen the 100 days that incorporated the peak of rainfall and the peak of net primary productivity, and also the sort of lowest 100 days around the lowest drought period. This was a study done by a colleague of mine, and he's found that there's a declining rainfall over the last 15 years, which is a little bit of a concern, because also drought frequency has increased on Aldabra. And this is a study that will hopefully be published next year, where we found that not only is drought frequency increasing, but also the response of the vegetation is changing. It's not recovering as quickly as it used to. So this might have large implications for the tortoises. So a little bit about the tortoises. There's 100,000 of them on Aldabra, which is quite a lot. And we, as I said, they're ecosystem engineers, but the best thing about them is that they have been there for, we think, around 80,000 years, and so the habitat we're looking at, we feel quite safe that they've been engineering this landscape for a very long time, which makes it great to study. But the other cool thing about them, if we want to look at how environmental or climate variables impact on them, their behavior, and therefore the vegetation, because they're ectotherms, we can look at different scales depending on whether it's large-scale movements or fine-scale movements, and then link those to the different types of variables that fit with those. So how do we do that? We attached 31 GPS tags to tortoises around the atoll, on particularly large tortoises, because the tags are particularly large. And they also contain accelerometers, so that allows us to get the large-scale movements, but also the fine-scale activity that we can get from the acceleration data. So first up, what we did is to calculate the utilization distributions. We do this through using Brownian bridge models, and this allows us to identify core areas of these home ranges. And we can take the whole home range, or we can take seasonal aspects of the home range. And hopefully from that, I was able to try and find some kind of seasonal difference. So once we calculated the home ranges, we had a look at this. So Picard and Grande-Terre have quite similar home range sizes for the tortoises, even though the islands themselves are quite different in size. And then we have the dunes coming next, where the tortoises have relatively small home ranges, and then Malabar, where they have tiny home ranges of about 300 meters squared, so not moving very far. There was a marginal difference between sex, with males having slightly larger home ranges than females. So we've got the home range size, but by looking at that and seeing we've got these two large home range sizes on the west and east, why is that happening? So in movement ecology, we like to classify movement, and we do that by calculating the net squared displacement, which is squaring the distance between the starting location and the end location of the path. And that gives us these nice curves, and then we run some models, and hopefully the models fit the curves, and then we can classify them into migration, resident, or disperser. So you might be wondering why I'm mentioning migration in a talk about tortoises, because people usually think that's to do with albatross or whales or these long-distance dispersals, but it's quite applicable in this case. And what you can see here is a classic migration NSD. You've got a starting point where it's quite sedentary for a while, decides at some point to leave, walks 6 kilometers inland, stays there for a couple of months, then walks straight back to the place it started from. Okay, and so from doing that, we were able to categorize most of the tortoises into one of three categories. And in a bit of context, this is what it would look like. So you've got a resident home range, quite small, quite contained, and then the NSD on the left underneath there, showing it's not really moving away from its starting location. And then on the right, you've got a sort of a migrator, where interestingly, this one didn't do anything for 2013, just stayed put, and then in 2014 decided to leave. So why would they do that? Is it because of the vegetation's changing? Is it because the season has not been accommodating for their body condition to be healthy enough? I'll try and explain that in a minute. But there was a couple of tortoises that we couldn't identify, we couldn't categorize. So we've now coined the term local migrator. I saw a paper the other day that called it exploratory resident, but the idea is quite the same. They're moving a lot, quite far distances relative for a tortoise, and they're not really staying anywhere in particular, and they do this on regular intervals. So they have quite large home ranges, but are not sticking to particular places. So if we try and look at the relationship between whether you're a migrator and what your home range size will be, you can see some migrators actually have home range sizes that are around the same as a resident or local migrator, and that's because you're having small home range sizes and this big movement inland to the same size home range in your other preferred area. So a little bit of context, and you have to be quite sharp. I know it's the end of the conference and everything, but the dots are going to be moving very quickly. So I've sped it up for you so it's not too boring. But yeah, so this is a video of their movements, and if you look on the blue one here, or the green one here, this is the migrators moving inland, and then you have a local migrator going back and forth along this landscape here, which is from mangroves into sort of dry habitat and back out to mangroves and back. And I think this is something to do with the tide that comes in through the lagoon. It's annoying it doesn't have a point. Anyway, so that's the tortoises on the east of Grand Terre. So then we try and, now we've done all that, done the home ranges, done the movement modes, movement strategies, we tried to link that up with the climate, the seasons, and to see what we could find. So here you can see the dry season. This is between sexes. You can here see the dry season, and they're quite similar. And then in the wet season, there's some differences there for the females, and then the males. You can see that big box plot with the male in 2013. So we were like, well, why would that be? And then we looked at the types. So you've got the migrating types surely have these large movements in the wet season in 2013, but in 2014 didn't do as much, which may be connected to the fact that in the dry season of 2013 was incredibly dry. So maybe the body conditions weren't good enough for them to move away from their preferred resource. So there was some evidence that seasonality played a role, but we couldn't really identify the exact drivers of what was going on. So we thought maybe have a look at vegetation per season, and again, there was not much signal. It's all quite similar, and a colleague of mine's working on a new paper to really get into the bottom of this. So we can sort of see that seasonality is playing a role in some of the movements, but as to the real reason why they're doing it, we're still uncertain. So we'd go from large scale down to fine scale. They're ectotherms. They need to thermoregulate to survive. Too cold, they can't operate. Too hot, they will die. So here you see the activity pattern. So we took a binary, converted the accelerometer into a binary, and then from that we get the red curve, which is the typical bimodal behavior of a tortoise during the day. Quite active in the morning, nothing during the middle of the day, a little bit in the afternoon. So we did this for all the tortoises, and we can clearly see that they're operating as we expected, and looked at over the year how that looks. And so the blue background is rainfall, and you can see during the year this bimodal shape changes, and in the dry season the afternoon activity completely drops off. And as this was averaged over all the tortoises, some were still being a little bit active in the afternoon, others were doing absolutely nothing whatsoever for numbers of days. So we can see clearly that there's some influence of rainfall, and that probably drives net primary productivity. So if this is just a mock-up of what it might look like when I actually do it. But if we take the NDVI data and plot that with the activity, we are pretty confident that we'll be able to find some sort of strong link there. So the next steps are going to be to use high-resolution drone data to pick up near-infrared imagery, and then we'll connect the movement paths and activity with that. And the cool thing about the tortoises is they're now being spread out to all these other islands which are functionally, how would you say that? They're not fulfilled in all their functions that require the islands to be restored, and so the tortoises are acting as analog replacements to extinct tortoises. And we think that using the data from this wild population in Aldabra, we can take that to islands in the Indian Ocean and help the organisations there manage their tortoise populations better. And in the future, what we'd like to do is try and predict their role in seed dispersal by taking the movement paths and gut retention time, feed them some seeds that we can track, and then hopefully they will show us how they do it. And with that, thank you very much.
Right, so thanks for the talk.
No worries.
Some time, so if we have questions for Richard, please.
Yeah, you kind of said it there then, but I was going to ask if there's things that we're going to be done in Mauritius, also have been introduced.
Yeah.
So we work with
Oh, yeah. Luca Berger from Swansea's running a project down there now. They have had GPS tags on there before, but that was just to get an idea about where they're going on the island. But now they've got these amazing daily diary tags. They're able to look at energy budgets, and how they're using the island. The island's very different to Aldabra. It's very steep, and they've already found that during the really hot days, they don't like to go up the incline, but on cooler days they do. So they're aware of how much energy they can be using in these times. So we will try and link up these islands. There's also Rodrigues as well has got a project similarly that we're involved with to look at the movement there and their effect on habitat restoration. So in the end, we'll be able to sort of try and scale up from very small scales to sort of Aldabra-sized. And then there's also a project in Madagascar where they're going to be introducing Aldabran tortoises to a ginormous site, and so hopefully we'll be involved in that as well.
Leading on from that, obviously maybe not obviously, but the ecological conditions on islands like Mauritius,
Mhm.
Areas, and
Yeah.
are really different from Aldabra.
Yep.
Densities are super high.
Yep.
To what extent do you think we can extrapolate from Aldabra to these other places, or do you think they would all look like Aldabra if they were, if they had enough tortoises?
Yeah, no, it's a good question. One thing about the density is we're still not clear. We know that if you constrain them in high densities, you're going to ruin your habitat. Like, there won't be any grass left. There'll still be some trees, but that'll be it. It would be dust. So you don't want to have them in too big a densities. But what we found on Round Island in Mauritius, where you have this sort of pinnacle island, they have been creating these little home ranges of where they've been eating the invasive grasses. So you're getting these nice tortoise turfs, these flat sort of savanna-like turfs. So I think, yes, you're going to end up with nice, diverse grasslands, and I think the other thing with the seed dispersal, how they actually operate is going to be different in each habitat. But they'll never end up looking like Aldabra, I don't think, just because of the size of it.
There was one more question.
Just briefly, I may have missed it, but
Yeah.
going back to some of the box plots you showed with the wet seasons in 2013 and 14,
Yeah.
there was a big difference between the migrators. And I just wanted to check if were they the same individuals. Did I miss that? Did you tag them for many years? Or
Yeah, it's a very long-term data set. No, you're right. I did fly over that. So the migrators, one thing we found that between tortoises is very variable, and within a tortoise it's very variable. So we still can't quite figure out what, and that's why I think that the 100-day periods that I chose to identify seasons, I think is probably the wrong approach, and I should actually go for like more of a monthly movement, and then try and capture that transition between when you're moving and when you're not, because I think we missed quite a lot of that, these transitions. But it's highly variable year on year. So this year the tags are still on, but I think they're finishing this year probably because the batches are dying out. Yeah, no, the data from that is fantastic. Yeah.
Well, thank you very much.
No, thank you.
Yeah, this is it. So thanks for sticking around.